the recording. And it'll probably ask you on your screen. I'm not sure how many of you have had experience with Zoom or not, but you'll probably get a prompt on your screen just letting you know that you are being recorded, and you can just hit the X and that'll go away from your screen. So I am going to share now. And can everyone see the screen okay?
Yes.
Okay. All
Clean
right.
Katie came to say hi. She's not
Hi.
going to stay for the whole session.
Hi, Katie. Did you have a good Christmas?
I am, yeah. How about you?
Oh, it's Brandy. Hi,
I
Brandy.
did, yes. Thank you. Happy New Year.
Happy New Year
Thank
to you. Yeah.
You too. Hi, Colleen.
Hi, how are you?
Good, how are you?
I'm pretty good. I have a cold, actually. Hopefully I won't start hacking in the middle of this. But
Oh, no.
so this is the first time, I believe, that we've offered this session virtually. And the goal here is just to make it easier for people to feel like they can communicate with one another. I'm happy to answer questions that people have. We do have some questions that have already come through via email, and so we will prioritize those. But if time permits, we're happy to answer any questions that you have. I will also send my email to Brandy. She'll be doing a follow-up communication with everyone, so that if you do have a question and just don't feel comfortable perhaps asking in a large group, or you would like to, maybe a thought came to mind that after the session that you would like to have additional information on, you can feel free to contact me one-on-one as well. For disclosures, I do want to note to everyone that I do volunteer as the scientific advisor for the International Mosaic Down Syndrome Association. And this is a group photo that we had from the conference this past summer that was held at Niagara Falls. I am with many of our self-advocates and family members. Also, I'm going to just give Brandy a second to say a few words. Brandy?
Hello. Thank you guys for joining us today. I think it was probably back in early March or maybe even April of last year, we had a campfire chat where we community chat. And one of the things that we had listened to was that people wanted to see more ways to be able to connect with Dr. Jackson Cook, and even having more webinars that pertain to Mosaic Down syndrome. So this is us trying to fulfill that. And so it's kind of been a little bit of a challenge to make sure that everybody's schedules and stuff work out nicely. So we started out this month that we were able to kind of get our schedules aligned. But yes, so that a lot of times we don't have to wait till we have a conference or the research and retreat. So this was just another way for us to be able to reach more of our community. So again, I'm happy that you guys have joined us today. We had about 62 people that signed up for this, so that's pretty exciting. And so hopefully we'll get to meet some new faces today too. But our conference is in Denver this year, so hopefully if you guys want to meet and connect with other families, that is a good opportunity to come down. And then obviously you would meet Dr. Jackson Cook face-to-face in a private setting or just in general, but that's another way to meet and connect with her as well. But yes, again, thank you guys for coming.
And as the session is ongoing, there is a chat window that you can type questions into, and Brandy's going to help me monitor that. Just trying to be efficient with your time and respectful of your time. We will try to keep the session to an hour and a half, no more than an hour and a half. So we may or may not have time to get to all the questions that people might have. But again, greatly appreciate your taking some time out of what I imagine is a busy time right now with the holidays and all. So with that, we'll jump right into it. So one of the questions that we received was, how does mosaicism happen? And the follow-up question from that same person is asking, is it present in the egg at conception or during cell division? And I think this is a great question. I thought I would start by simply defining what is mosaicism. And so the scientific definition for mosaicism is an individual that has two or more distinct cell lines that originate from a single zygote, but they differ because of mutation or non-disjunction. And so I'm going to unpack that a little bit more in the next slide. So, how does mosaicism happen? There are two ways that it can happen. From our studies, as well as studies of other researchers, the most common way it happens is that it starts off with a change that occurred in a gamete, so either in an egg or a sperm, most often in an egg, where there was a meiotic sorting problem. And then once that egg is fertilized, it results in a trisomic zygote. So you could have the problem in the sperm or the egg. The result is that you have a zygote that has the initial cells are trisomic. And then as that zygote or early embryo undergoes multiple cell divisions, then there's a second chromosome sorting problem that gives rise to that second cell line. So that one of the chromosomes 21 is lost. And so now we have some cells that are represented here with blue that are disomic, or two chromosomes 21. And then the second cell population that's trisomic, or has three 21s. And then these cells will continue to proliferate, and that's how you get the two different cell lines that arise. The other way that this can happen is that you can start off with a zygote that has the typical chromosome number, or two chromosomes 21. And then during some of those divisions, as the embryo is developing, it might retain an extra copy of chromosome 21 to give rise to the trisomic imbalance. This is another way of looking at that same information. So we start off with a cell that has one chromosomal makeup. That cell will divide to make two identical copies of itself. And then at some point along the way, this is showing it early in the divisions, but it could happen anywhere along the division process. You might have a chromosome sorting change, so that, say, if this started off trisomic, then this cell might have a disomic, or have two 21s. The other cell, the daughter cell, might retain those three 21s. And then those can continue to divide, so that you have some cells that have three 21s and some that have two 21s. Another question that we were asked is, what is mosaic translocation Down syndrome? And is this significant in any way? And again, another great question. So when we look at non-mosaic Down syndrome, or trisomy 21 is the most common chromosomal makeup. So about 95% of all people who have Down syndrome will have a total of 47 chromosomes, where they have one extra copy. Typically, we have two copies of each chromosome pair. One of those chromosomes we get from our mom in the egg, the other comes from the dad in the sperm, and then we reestablish that number of two. But in individuals who have Down syndrome, they have an extra chromosome 21. So instead of 46 chromosomes, they have 47 chromosomes. But there are also other ways that you can have symptoms that are associated with Down syndrome. And one of those ways is what's called a translocation. And this is seen in about 2 to 4% of all people who have Down syndrome, is that they, instead of three 21s, they'll have what's called a translocation. And so in this case, when you count the number of chromosomes they have 46 chromosomes. But as a cytogeneticist, what we do when we diagnose patients and look at their chromosomes is we go band by band and we compare all the stripe patterns. And you can see at this point, this chromosome has a lot more material on the top than this chromosome. And if you imagine this with a break and then flipped upside down, what it is an entire copy of the long arm of chromosome 21. So there's 1, 2, 3 copies of chromosome 21. This is a schematic that shows it in the way that you can have exchanges between 14 and 21. so that the 21 gets attached onto the 14. This is what we call a 1421 translocation. This is a shorthand way, sort of a hieroglyphics way that cytogeneticists describe what we're seeing at the microscope and with our imaging tools. It has a rearrangement between 14 and 21. That's what this first parenthesis is telling us. The second parenthesis is telling us that there's a long arm of 14 that's fused with the long arm of 21. But we also have two free-lying 21s, so we have an extra dose of 21 compared to expectations. This is one of the more common types of translocations you can see. Another one of the more common types of translocations involves a 21-21 rearrangement. And so in this case, again, there are 46 chromosomes, but now we have 121 with a second 21 on top. So we have 1, 2, 3 doses of 21. So this is, again, another type of translocation. And there can be other forms of translocation besides these. These are two of the more common types. The question also asks, what is the significance of having a translocation? As far as outcome, there really is no clear difference in the health or development expectations of someone that has one of these types of translocations. The real importance in knowing if someone has a translocation is that sometimes you can have a parent who will be a balanced carrier, and so that can impact their recurrence risk. It could also impact the recurrence risk of the individual themselves who has Down syndrome. So that could be another reason that it's important to know. There are some other types of translocations that might involve only parts of chromosome 21 being present in 3 copies. And in those cases, it can impact the outcome in that since it only has a portion of 21 in 3 copies, usually we tend to see atypical presentations, sometimes milder outcomes, but not always, depending on what the rearrangements are. We talked about translocation, but there's also then, you can couple that with mosaicism. So in this case, you might have some cells that would have this chromosome makeup, whereas there could be other cells that would have just the 2 chromosomes, 21. So that's combining this mosaicism category and the translocation category. One way that we can quantify or measure the percentage of trisomic cells is using a technique that's called fluorescence in situ hybridization. And here what we do is to take a piece of DNA and we highlight it with a red color or a green color. In this case, it was a red color. So we see 1, 2, 3 copies in this cell, 2 in this. So this would be a trisomic cell. This would be a disomic cell. The green color here is for a different chromosome. It could be chromosome 8 or 21. If we look just at interphase cells, we can't tell if those 3 copies are due to a translocation or trisomy. We have to actually look at the chromosomes to determine if there's a translocation. So that's why, even though we have all the new high-powered technologies for diagnosing a trisomic imbalance, it's still very helpful to use chromosome testing so that we can see the structural changes that are associated with Down syndrome. Another question I have is, are some types of cells better correlated to phenotypes? And again, y'all had really good questions. I was very impressed with the questions. So to answer this, I thought it would be helpful to go back to thinking about embryogenesis again. And so we start with an egg that's fertilized by a sperm. And once we have that fertilized egg that's formed into the zygote, that will go on then to make cells that will ultimately end up forming the placenta, as well as the amnion or the chorion. And some of those cells will go on to make the embryo. Within that embryo, then these cells will divide into different layers, and those are shown in more detail over here on the left-hand side of the image. And so there are three main layers that the embryo will form. One is what's called ectoderm, shown here in blue. And this is what will ultimately go on to form the skin, the central nervous system, and other tissues. We also have endoderm that's highlighted here in gold. And these will go on to form the lining of the gut, the liver, the lungs, the pancreas. And then the third layer is what's called mesoderm. And this is what forms the bones, cartilage, muscle, the heart, and blood. And so when we do our research studies on people who have mosaic Down syndrome, we like to look at both blood and buccal cells. Buccal cheek cells, or buccal mucosa is a scientific word for the inside of the cheek cells. But those cheek cells represent the ectodermal layer of the embryo, whereas the blood represents the mesodermal area. At this point, we haven't sampled cells that come from the endoderm. We have thought a little bit about how to do this, and because all these, as you can see, are internal type cells, the only way I can think of to do that would be potentially to get a stool sample and then to extract cells from that. But there are challenges with collecting that, so we haven't gone there just yet. So the question about how does it correlate then with phenotypes? When we're thinking about mosaicism, we have to think about when these changes happen. So we have this chromosome sorting problem. If this happens very, very early in the embryo, then all three of these layers will have that mosaicism present. It's also possible that you can have the chromosome sorting problem that happens later, excuse me, in embryogenesis, so that it could be just one layer that might be affected versus the other layers. Or you might have it happen early, but then the percentage of cells that have trisomy don't get split equally between these layers. So it's actually a very complicated question, and one that we've been trying to look at. That was one of the goals that we had when we requested looking at blood and buccal specimens from our individuals who have mosaicism. We have seen that there are some differences. Some of those differences are present at birth, but some of those differences between different tissues change with aging. And that's something we'll talk about a little later in this hour. Sorry, as I said, I've had a cold, so I'm trying to avoid coffee. Another related question that I had is, are there karyotype and phenotype correlations for mosaic trisomy 21? And how strong are these correlations? So one thing, as I said, we've done with our study participants, we get information about their phenotype, and this comes from review of actually looking at the individual with physical exam, getting medical records from the individual. We have a questionnaire that we ask the individuals and or their parents to complete, and also photographs. We also then collect blood specimens as well as cheek rubbing specimens. We look at 1,000 cells from the blood specimen, 500 nuclear cells from the cheek specimens. We compare the percentage of trisomic cells in each of these specimens, and then also compare those percentages with the outcomes. And what we've seen is that in general, in our study participants, looking at all the participants, so we had matched buccal cheek cells and blood cells, the percentage of trisomic cells in the cheek cells on average was 46.06%, plus or minus the standard error is 3.44%. In the blood, that percentage was 30.13%, so lower in the cultured blood. One question we had was, well, maybe it's the culturing process, because the cheek cells we don't culture in the lab. Maybe there's something that's selecting against the trisomic cells in the culture. So the other thing we did was to compare the percent of trisomic in the cultured blood cells to uncultured blood cells. And here we saw no significant difference. So that suggests that this variance that we're seeing between the blood and the cheek cells is a true biological variance. When we look at correlations between IQ scores and the percentage of trisomy in the cheeks and the blood cells, what we find is that there is a significant inverse correlation between IQ scores and the percent of trisomy in the cheek cells. So in other words, the higher percentage of trisomic cells that we saw on the cheek specimens, those tended to have lower IQ scores. The cheek cells had a significant correlation. And this value was also more strongly correlated when we compared blood. While we saw a trend, it did not reach significance. So at least in our data, the cheek cells tend to have a stronger relationship with performance, academic type performance as measured by IQ, which in and of itself has some limitations. That is especially interesting because when you go back and look at embryogenesis, you'll remember that, there we go, that the central nervous system or the brain and the buccal have the same embryonic origin. So it suggests that there are some differences that can be detected by looking at different types of cells. Similarly, when we looked at the correlation between congenital heart disease and the percent of trisomic in cheek cells and blood cells, we saw a closer relationship with heart disease and the lymphocytes compared to the buccal cells. And both the heart and blood have the same embryonic origin. So short answer is yes, we do see some differences in outcomes with traits that are associated with Down syndrome by looking at different types of cells. But these are not so strong as to be predictive at this point. So for example, I can't, by saying that you have 60% trisomic cells, give you an accurate prediction of how well an individual is going to do with their outcome. In general, we find that people who have lower percentages of trisomic cells do tend to perform better than others. But again, that depends on, have you looked at both the cheek cells and the blood cells? I think the cheek cells tend to be more constant over time is what we've seen. And so those might be a better predictor. Having said all that, you can take two individuals who have 50% trisomic cells and they can have very different skill sets. So yes, we see a relationship, but at this point, that relationship is not strong enough to be predictive. Another question that was asked is, does the estimated percentage of cells from an amniocentesis karyotype predict that most other tissues after birth would have relatively similar percentages, or can there be variation in the percentage of affected cells across different cell lines? And this is a great question. Most of the time, we do see similar percentages at birth. Those percentages can change with aging, but it is possible that you can see differences in different tissues. And so the follow-up question to this was, what do you recommend for testing to confirm a diagnosis after birth that could give the best representation of how many cells are affected? And what I would recommend following birth is that a blood sample be collected as well as a cheek rubbing specimen so that you get two different tissues. Sometimes individuals will, if the birth hasn't happened yet, You can plan to have skin cells collected from the umbilical cord, which would be a skin type specimen. And that can sometimes be helpful or informative also. The amnio, the tissue, the cells that are in the amniocentesis specimens are most like skin cells. So we would expect to see a closer relationship between what was the amnio result and the results that we see in the skin-like cells. Is there evidence about the types of cells that should be used for testing? And so we've got several questions here that are related. There are strengths and differences, strengths and weaknesses to each as far as the types of cells. Most people, when they are first having their diagnostic test, the first specimen that's collected tends to be blood. And that's a really good starting specimen because what the blood does is to enable us to actually look at the chromosomes. And so we can distinguish not only the percentage of cells that might have a trisomic complement, but we can also determine, are those cells, do they have 3 separate chromosomes, 21, or might there be a translocation present? So that blood sample is really very, very helpful. The advantages of doing blood with our typical G-banding type approaches is that you can identify the translocation. You could also do that if you had a skin biopsy, but this is more invasive. Typically at the initial diagnosis, they're going to evaluate 20 to 50 cells. And there's been a little bit of misinformation out there about how many cells do you have to look at to diagnose mosaicism. 20 to 50 cells, 95 to 99% of the time will pick up mosaicism if it's present. And these values were determined based on the probability that if you had, so by looking at 20 cells, if you have a trisomic complement that's present in 10% of cells, there's a 95% chance that you'll pick it up by scoring only 20 cells. So it's a probability estimate. Having said that, there are some individuals that do get missed. It's not often, but there is some potential. But most of the time, 20 and certainly up to 50 will be sufficient to pick up that mosaicism. And we have special rules as diagnostic cytogeneticists that we have to follow to make sure does this meet the criteria of mosaicism. Probably the most robust way for picking up mosaicism is doing that technique I've talked about earlier, where we look directly at cells and then we label part of the DNA with a color. And then this technique, we can score more cells very robustly. So typically people will score at least 100. In a research setting, we score 1,000, but that's not, it's really just for research to get an extremely robust measure. 100 would be adequate for detecting mosaicism. A beauty of FISH is that you can make this assessment on any type of cell that has a nucleus. So you can use cheek rubbing cells that are not invasive as a sample to look at. There are a lot of different cells that could be used. Some of you may have had, depending on the age of the individual who has mosaicism, you may have had a microarray test. This is a technique that has higher resolution than our standard testing. Depending on the lab, in our lab, we're able to detect as low as 4% trisomic cells with a microarray. But again, FISH just has a higher ability to pick up those trisomic cells. And then next generation sequencing is another type of diagnostic test that has the potential for picking up those percentages. Neither the microarray or the next gen sequencing, while they have higher resolution, they really are measuring just the amount of DNA. You still need the G bands to determine is there a translocation present or not. Along the question that you ask, is there evidence about the different types of cells for testing? When we look at our research data, and look at the frequencies of cells that we've seen that have trisomic complements over time. So these would be any person that has the same color and symbol would be one individual that we've studied over multiple different time points. And what I don't have here, and we need to gather for maybe the next session, is what was their percentage of trisomic at birth. This is the information that we had from looking at blood cells. These are matched cheek cells, so the same individuals in both. And you can see, for example, this individual that has sort of a gold color here has a pretty consistent percentage of cells that are trisomic, but we have lower percentages in the blood. You can see just the patterns are spread out much more over different percentages, with many of them hovering around the middle point with the cheek cells, while as in the blood, the values are lower. And so what we have seen in our individuals is that over time, the blood cells will tend to have a lower frequency of trisomic cells, but the buccal frequency tends to stay pretty constant. These lines are pretty level for the most part and not statistically different over time. So that's an observation that we're continuing to do research on and to try to better understand. Again, the constitutional at birth percentages is going to mirror pretty much what the cheek rubbing data shows. And this is just showing the average changes in the of trisomic cells over time. And you can see that with blood in general, the percentages over time. So this is looking at differences, taking the initial and then looking at that difference. And we see generally a pattern of decreases in the percentage of trisomic cells, but not huge differences. Similarly, with buccal, we see a pattern of about the same or potentially some modest gains, but no big differences.
Colleen, real quick, would that answer the lady's question? Can the percentage of mosaicism change lower or higher percentage over their
lifetime? Yes. So that's a great question. And yes, when we look at blood, we can see the percentage of cells that will change over time. So that's something that, and I'll talk a little bit more about that as this hour continues. Why is that? Why are we seeing these changes? The buccal values will tend to be more consistent across time. The real question, and again, I'll talk about this a little more at the end of the session, the real question many of us have is what's going on in the brain. And most of the brain cells are not actively dividing. So this change in the percentage of trisomic cells seems to be related with the cell division process. And so we would expect that the percentage of cells in the brain will stay fairly constant throughout the lifetime. But that's, again, something we can't really measure because you can't non-invasively assess the brain. Another question we received is, can the non-invasive prenatal test, abbreviated non-invasive prenatal test, detect mosaicism, and can it detect all percentages, both high and low? And again, this is a great question. I wanted to start by answering, there are different types of diagnostic assessments that can be done. One is what's called a screen, which is an assessment that's completed to determine an individual's risk for having a particular condition. The other is called a test. And this is an actual diagnostic assessment to determine if the person has that specific condition. The NIPT screen is that it's a screen. It's not a diagnostic test. So it defines is someone in a high risk group or a typical risk group. The non-invasive prenatal screen is done from taking a blood sample from a woman who is pregnant. And then what they're doing is looking at the DNA that's in the maternal circulation, but then also looking at DNA that comes from the placenta. So it's coming from the placenta rather than the baby itself. And then they're looking at the counting when the cell-free against the maternal screen. And so if you have a baby that's carrying an extra chromosome 21, for example, If there were equal amounts, you would expect to see equal ratios of maternal and fetal DNA. If there's an extra copy, then you see a shift of those ratios. And so the way the screen works is that they will actually detecting DNA values and then counting those sequences based on the expected number of sequences you would have. They align those to the chromosomes. And these are slides. This slide is from one that is cited here by the American Journal of Obstetric and Gynecology. This is one that's from a textbook that we use for teaching. One uncommon but sometimes detected change that can happen with non-invasive prenatal screening is that, again, you can have a situation that's called confined placental mosaicism. So we're not, in the screen, you're not sampling the baby directly, you're sampling the placenta. And so there are some situations where the placenta may have a trisomic imbalance, but the baby would actually have a typical chromosomal makeup. So this would give you a high-risk non-invasive prenatal test, but if it had a follow-up amnio, you could see a normal result on the amnio. Another type of confined placental mosaicism is that the placenta may be normal, and then the baby could have a trisomic complement. So this would give a low-risk non-invasive prenatal screen that could potentially miss then a child that would have a trisomic imbalance. And then the most common type is that both the placenta and the fetus would have the same chromosomal makeup, and so it would be correctly categorized as high risk for having a trisomic imbalance. Looking at confined placental mosaicism, again, there are different ways that this can happen. You could have an embryo that starts off with a trisomic complement. We then have what's sometimes called trisomy rescue to give rise then. So the trisomy is focused only in the placenta. The fetus has 2 chromosomes, 21. And so that's where you have this discrepancy. And this is just going over that same information. You can have starts off disomic, but then you may have some gains that are localized only to the placenta. So there can be differences between the placenta and the baby that can lead to some discrepancies in the non-invasive prenatal screen. But most of the time, that non-prenatal or cell-free prenatal screen is actually a very good screening test to detect the more common chromosomal changes, especially trisomy 21. A negative result does not fully rule out the possibility of having a baby with a chromosome condition. If you have a positive result, it is strongly recommended that a diagnostic test be determined to look at the fetus directly. And so you can see, we recommend that all couples first have consultation so they fully understand what the screen will provide. If there's a positive test, depending on whether they had that screen done, usually it's done no sooner than 10 weeks, anywhere from 10 weeks or later in gestation. And depending on when that time is done, have either a CVS or a first trimester or second trimester prenatal test done if it comes back positive. If it comes back negative, then typically there's no further testing that would be needed. I see in the chat there is a question, would I recommend getting a buccal specimen done if a blood specimen was only done at birth? If you're looking for mosaicism, if that's your real question, if there's no hint of mosaicism that's picked up at birth on a blood specimen, most of the time that individual will not have mosaicism. We have had some individuals who had a normal result at birth, but had many of the traits that you see associated with Down syndrome. This is something that the IMDSA has sponsored, what we call our cryptic mosaic study, where we have looked at some individuals who have had screening done, and we have identified a couple individuals with buccal specimens who had trisomic cells by looking at cheek specimens that we were not able, that their testing center was not able to detect in the blood specimen. But for the most part, it's that initial diagnostic test is going to be an accurate result. We look at the buccal really more from a research perspective. It's not really going to tell you, if I'm seeing the blood percentages change, it's not going to change how you manage the health of your child by having both specimens done, but it helps us to understand more about how the cells are growing and looking for potential relationships that might help us to better understand aging changes that might happen. Another question we got is, what is the current research on reproductive organs for people with Mosaic trisomy 21? And can they conceive? Again, another great question. The can they conceive question I'll start with first. This is not a one-size-fits-all answer. Individuals who have non-mosaic Down syndrome, for the most part, most males who have non-mosaic Down syndrome are not fertile. There have been some exceptions to that. We certainly have examples of males who have mosaicism who are fertile. So I think, that's something that you would assume there's a likelihood that they could have fertility. The checkpoints in spermatogenesis versus oogenesis are different. They're more rigid in spermatogenesis. And so if you have differences in the chromosome number, that will cause spermatogenesis to halt. And that's why in males you see more infertility in people who have Down syndrome. Females, even those who have non-mosaic Down syndrome, are fertile. And certainly females who have mosaicism would be expected to be fertile. What then is their risk for having a child with a chromosome change, just like them, or having a child with trisomy 21? This is the schematic that kind of walks us back through. You may remember learning about meiosis, or that special cell division that happens in eggs and sperm. So I'm showing just here the chromosomes 21 in a cell that has 2 chromosomes 21, or your typical makeup. Normally what happens is in this first division in the gametes, or what's called meiosis 1, normally those gametes the chromosome bivalents would pair, and then they would separate from one another in what's called the reduction division. And then in meiosis 2, these replicated chromosomes will separate so that you would end up with a gamete that has only 1 chromosome 21. If this was in sperm, for example, half of them would have the homologue that's shown in blue, half would have homologues that are shown in pink. If we look at trisomic cells, now instead of having just 2 chromosomes to sort, we now have 3 chromosomes to sort. And so when this happens, the most typical way that this happens is that 2 chromosomes may go to one pole, with 1 chromosome going to the other pole. When those continue to divide, the replicated chromosomes divide. The cells that came from this portion of the division would have 1 chromosome 21 and could result in a typical gamete. These would result in a gamete that has an extra 21. So for a person who has 100% trisomic cells in their gametes, their risk for having a child with trisomy 21 is 50%. Half of their cells would have the extra 21, half would not. If we have a person that has 50% trisomic cells, then their risk for having a child, the typical cells, you would not expect to have them. Half of the trisomic cells would result in a normal complement, half would result in a trisomic complement. So their risk, if they had 50% trisomic cells in their gametes, would be approximately 25%, and so on. The real challenge here is knowing what is the percentage of trisomic cells that the individual has in their gametes. And that's a case where it could be helpful to look at blood and buccal cells to know Are we seeing consistent percentages across all different tissues so that we might expect to see that same percentage in gametes, or are we seeing differences? The other thing is that these risk calculations are based on someone who has 321s in their trisomic cell. If we're looking at a person who has mosaic Down syndrome with a translocation, then that risk can vary depending on the translocation. Are there any questions on this? Or anything we've covered so far? Okay, I'm going to keep going. Another question we got is, are there trends in areas of development? For example, is it common for delays in speech? And so if we look at the individuals that we've studied who have mosaic Down syndrome and compared their traits to those we see in non-mosaic Down syndrome, what we see is that many of the traits that you see in non-mosaic Down syndrome are observed in people with mosaicism and in comparable percentages. We have detected some traits that we see less frequently, for example, the upslanting eyes, the hyperextensibility of joints, congenital heart disease. We've seen some decreases in those frequencies among people who have mosaicism compared to non-mosaic individuals. And there are some other traits that we tend to see some differences in. Interestingly, leukemia, we see at a not statistically higher, but numerically a little bit of a difference there. If we look at learning challenges that we see, many of these challenges really are very similar between people who have mosaic compared to non-mosaic Down syndrome. Some of the more common areas for learning challenges, and again, every individual is different, just like people who have typical chromosome makeup. Some people have talent doing math, some people have talent doing art. Individuals who have mosaicism are going to be the same way. Yes, you have some cells that have that extra 21, but you've also got thousands and thousands of other genes that reflect the rest of your makeup. And so if you might have parents who are both talented in art, maybe that individual will have a propensity to do art well. So each case is an individual, and you really have to look at what their talent levels are and their challenges are. But overall, as a general theme, verbal memory seems to be a challenge for many people who have Down syndrome or non-mosaic Down syndrome. So they tend to do better if you give them instructions, written or pictorial instructions versus verbal instructions. Attention spans can be a bit shorter. Expressive language can be a challenge for people with Down syndrome or non-mosaic Down syndrome, so that they do have slower development of speech and conversation skills. This often results in simpler sentences. Processing speed can be a little slower paced compared to their peers. Motor skills also that can lead to them having some delays in acquiring the skills for handwriting, and also hypotonia. That's why for young babies, we'll often recommend infant stimulation to exercise those muscles and help them to develop that better muscle tone. Some learning strategies that some people have found to be helpful, using visual aids for teaching. So using pictures or charts. Visual aids oftentimes are easier for the person to interpret than just listening to instructions. Giving short, clear directions, so just using simple sentences, not overloading the person, not complex multiple instructions at the same time. Repetition and review, so revisiting skills that have been mastered to reinforce them in the long-term memory. Multisensory approach can be helpful, so both combining visual and auditory as well as hands-on methods. And then just individualized plan, where you focus on each individual's unique strengths. So that is something that really for all people, identify where their strengths are, where their areas for opportunities are, and really focus on optimizing those regions. Another question that was asked is, what can one anticipate to expect as far as intellectual and physical disability for a person with Mosaic Down syndrome? And the person who asked this question, I'm afraid I'm going to join the chorus that they've already heard, is that we really can't predict. Just like with any baby, if you have a baby who has a typical chromosome makeup, we can't really predict how well they're going to do. Just looking at our group of individuals who have mosaicism, we see huge variability in the skill set. Some of our individuals have many of the challenges that are completely aligned with expectations of someone who has non-mosaic Down syndrome. Other individuals are doing extremely well to the point that it would be hard to know that they had mosaicism unless you had that knowledge in advance. So there's a wide range of outcomes that one might expect. We have a small group of people who were diagnosed prenatally, and we have seen that those that had very low percentages from a prenatal diagnosis, many of those individuals went on to do quite well as they became older individuals. When they're diagnosed postnatally, it's usually because there is some sort of a health or developmental concern that brought them to the attention of the healthcare. So there's a little difference in the biases that are leading to the diagnosis. So kind of dancing, I guess, around the answer, but the reality is we just can't with confidence predict. Brandy, anything you'd like to add to that? You're muted.
Basically just what you said. And I think I've said this before. I mean, I have two sons who are typical developing, and I thought maybe one was going to be a lawyer and the other one might have been a doctor, and one's a teacher and the other one is. So you really just don't know what the future holds. It's hard to really predict the type of course that, the individual is going to go, especially, with what chromosomes are affected. But I will say this, no matter what, no matter how many chromosomes are affected, it doesn't matter. Continue to educate, continue to seek all of the resources that are available to you, whether it's your local school or the local disability community within your community in your backyard. We had a thing that was called Help Me Grow. They came out up until my daughter was 3, and then she went on to the school, and then we still got speech. Anything that you can qualify for, do it. And at the end of the day, they could say, you know what, they've outgrown it. We've already climaxed and we're at, fine, great. Continue to read to them, treat them like any other child. And I think that's, to me, was the biggest takeaway. And I've done that. And so my daughter's, 19, and I feel like she's in the middle of the spectrum. Because I like to think that this is kind of a wide range of cognitive ability, and it's like a spectrum. So again, I feel like she's in the middle. This past conference was the very first time she's gone to all of the conferences, and I've been with the organization going on 18 and a half years. And this was the first time that she actually actively participated. And I thought she did pretty well. She actually was up on the panel, I believe, with John and Katie. And so they got to, she got to share the stage. And so she was able to answer some of the questions appropriately. I think some of the questions might have been a little over her head, but again, she's, like I said, some of the stuff that was kind of career and long-term goals. So again, it's really hard to anticipate and expect what's going to happen. Just take advantage of all the resources and continue to educate them just like you would do any of your other kids.
Again, all the questions were just really good. I was very impressed with the questions. One person asked, are there estimates as to how many individuals with Mosaic Down syndrome are never diagnosed? And if they aren't or if they're misdiagnosed, what might they be categorized as having? Um, I'll approach it first the other way around. About 2 to 4% of all people who have Down syndrome are thought to have mosaicism. Um, as far as how many people might there be walking around who have mosaic Down syndrome that have never been diagnosed, we don't know. Certainly it is possible. We do know, for example, and this is worldwide and nationwide, some people, and even within our own group, some people have learned about their diagnosis because they developed leukemia. And that was the first time that they learned when they had their chromosome testing done for their leukemia workup. They identified the trisomic imbalance and then went on to do the constitutional studies. So, yes, there are people that are out there. There have been parents of children who have trisomy 21 that might have multiple children with trisomy 21 that's not translocation. They have no phenotype. Then when they tested different tissues, they were able to pick up some low level of mosaicism. So can it happen? Yes, there are clear documented occurrences where people can have mosaicism and never be diagnosed. We have no real, strong estimates of how likely that can be. We know that all people, all of us, as we age, also acquire chromosomal changes. In general, women tend to lose their X chromosome, men tend to lose their Y chromosome most frequently, but we all acquire changes as we age. And so, the other part of that question is it kind of depends on what the age of the individual is that you look at them. And if it's a chromosome change that happens after all the organs have been formed, then that really doesn't have the same impact. It can still be associated with health conditions, but it's not going to change the way the organs are formed. So it's actually a great question, but a pretty complicated one. So I hope I didn't go too deep in the weeds on that one. But another question is, are there new developments or research for older individuals who have mosaic Down syndrome? And what opportunities are there for development in jobs and careers? And I can say that aging in general, so one of the success stories of research with Down syndrome is that we have seen an increase in the age that individuals with non-mosaic Down syndrome are now able to live to older ages, but that age is still reduced compared to someone with typical chromosomes. And so one area that we're starting to focus on now that our people are living into older ages is, what is it like? What are their age-related risks? And what we've seen is that there are people who have Down syndrome or mosaic Down syndrome tend to have precocious aging traits. So they'll tend to get, for females, they'll have menopause at an earlier age. For people with Down syndrome, this is, again, data from non-mosaic Down syndrome, but menopause will start at the age of 47 in those women compared to 51 average in the general population. We see cataracts at a much higher percentage at this comparable age range. Hearing loss occurs at a much higher frequency in people with Down syndrome. Osteoporosis at 18 or older, that'll start showing up in about half of the people with Down syndrome. Of course, Alzheimer-like dementia is something that we see more frequently in people with Down syndrome. Diabetes are, again, some of the age-related traits that we see in people with Down syndrome. Solid tumors, interestingly, has a lower frequency in individuals who have Down syndrome compared to the general population. So it's one of those things that, yes, the health is a big concern, and we're trying to understand, why do we see these health changes or increased risk? A lot of it seems to be related to inflammation, but we're learning more and more about that. The second part of the question, I think there's a huge need, is opportunities for the development of jobs and careers. I think many of the parents of older individuals were the pioneers who trailblazed getting improvements in our school system. They're still not perfect by any shake of the imagination, but huge improvements that were made by that generation. And so now I think the next real hurdle that we have is developing more career opportunities and more job opportunities to help people who have mosaic Down syndrome or non-mosaic Down syndrome even give them opportunities for career development and job chances, and for assisted independent living. So these are all challenges. For jobs and careers, just looking at our people who have mosaicism, again, it goes back to what are their skills? What do they do best? We have, some people who will do, we have one individual who's a welder, for example, and he's done very, very well in welding. We have some people that do clerical work. It just really, some that are teachers' aides or assist with teaching. It really comes back to what is their skill set and what is their greatest talent. Anything else you want to add to that, Brandy?
Yeah, currently I'm in this stage of life right now with my daughter, and I kind of just base it on her personality. She's more of a quiet, more, I would say an introvert kind of person. And so she doesn't really want to be on the forefront of, trying to make people happy. So right now, the different career paths that we have that are through the local, like Butler Tech, it's just a next step after high school. And so we're looking at maybe her working at the local college or the local hospital, and we're kind of filling that out. And so she would probably work behind the scenes, like more in the cafeteria, and she seems to kind of like that. There's another part that she could work in the library up at the college. And so far we're just kind of, right now we're working on her just standing longer than an hour. That seems to be a challenge for her because she gets very tired, especially if there's not a lot of stuff going on. She tends to just want to sit. So these are the things that we are just kind of working on. So hopefully that as you go through high school and stuff, that you will start working on those things and getting those going and lining up and stuff and seeing what's available within your community. Again, I can't stress it enough to always tap into your local resources.
And I'm going to segue over to the chat a little bit. One question is, has the funding been impacted by the federal cuts in medical research? Not yet, but it is an area of concern. For the upcoming grant, I submitted a new grant with a collaborator in Florida. And of course, that grant was supposed to have been evaluated in November. That got postponed until January when the government was shut down. I have heard that they're not funding any new grants in the coming cycle because of the lack of federal funding for NIH budgets. So at this point, I do still have my funding, but I am concerned. I think it's an area that we all need to be concerned about. Global and others have been very active in trying to keep the Include project approved for funding. But again, if the budget doesn't get approved to appropriate money for NIH, then that isn't going to happen. And the priorities at NIH have definitely shifted with the current administration. So I am concerned. Other questions we have is individuals who are based in the UK and wondering about research collaborators there. And so I jumped to this question because this young lady, well, 46-year-old, I guess I think of her as a young lady, but she's from England, actually. She comes to join our conferences every year. There are researchers in England. You can also, though, participate internationally. We send kits to people. If you're in England, would like to participate in our research, we can make that happen. But there are some great groups in England. There's one group in particular that's one of the leaders in the world as far as looking at behavioral conditions associated with non-Mosaic Down syndrome. They're not focused on Mosaic, but they are among the leaders in the world on non-Mosaic Down syndrome. So yeah, there are some great resources there in the UK, and happy to talk offline about that as well. And I see there's another person here that sounds like there's some connections that can be made from this. So is it okay if I go an hour and a half?
Yeah, I'm good with that.
Okay. So another question I got is and for those of you who can't commit more than an hour, that's totally fine. Don't feel like you have to stay, but I would like to try to get through the questions as best I can. So one question is there any research on the function of genes on the 21st chromosome that are overexpressed? And does this lead or assist in our understanding of illnesses that a person with Down syndrome might be predisposed to? So one thing that we've been doing in our research group on people with Mosaic Down syndrome is that we have been studying single cell gene expression patterns in the genetically identical, except for the presence or absence of the chromosome 21, or what we call isogenic cells, and then looking at those changes with age. And so what we've been looking at are what are called single cell transcriptome studies. So what this is a fancy word for saying, what are the proteins that these, what are the genes that are expressed in these cells that will ultimately go on to form proteins. And so what we do is to look at the disomic compared to the trisomic cells. And then we look at the different clusters of cells, look at regulatory networks. And then we do a lot of high-level computerized bioinformatics studies. And so what we found in our preliminary data, we have some samples that we just sent from the last conference that I'm hoping to get those results back in the next few weeks. But from the preliminary proof of principle studies, one thing that we've seen across all individuals that we've studied so far is that if you look at the gene expression changes and look at the chromosomes that are involved, we can see the most changes are localized to chromosome 21, which is what you would expect. But we do also see some changes that can occur across other chromosomes. So that means having a trisomic imbalance not only affects genes that are on chromosome 21, but it can also affect genes that are on chromosome 20 or on chromosome 14 or chromosome 2. So it's not a straightforward just one chromosome sort of scenario. If we look at the trisomic versus disomic gene expression patterns, what we're seeing in this volcano plot is that there are some genes where we have overexpression, and many of these are very interesting genes. Many of these are genes that are associated with inflammatory changes or inflammation changes. Amyloid precursor protein is one that we would expect. It's localized to chromosome 21 and has been associated with Down syndrome and Alzheimer's risk. So there are a lot of different genes, the ones that we're seeing. So these are the genes that when we compare the trisomic to the disomic cells, which ones are different in the trisomic. And so this approach of comparing disomic and trisomic cells really gives us a clear advantage of taking away all the background noise to really see what is the effect of a trisomic cell. And so what we're doing first of all is looking on an individual level to identify the genes that are expressed more often or expressed less often. And then we'll look for commonalities across individuals to see is there something that's unique or specific. What we're seeing, again, as a whole, when we look at our trisomic and disomic cells, is that we see both types of cells in a multitude of different cell types. Inflammation pathways seem, the genes that are involved in inflammation pathways we see are differentially expressed, genome-wide, not just 21. We've seen mitochondrial genes that are affected, as well as some genes in senescence pathways. Some of our future work is to look at how do these change with age. So we have individuals who have participated in our study more than one time. And so we can look at what were the differences at age 13 compared to age 20, for example. And then also looking at how those changes are related to other aging hallmarks. And then another question I got is, what are you planning for future research? And I'll just give a very quick overview here. We have the non-Mosaic repository and Mosaic and non-Mosaic karyotype repository. This is mostly our individuals who have Mosaic Down syndrome that we've studied over. I've been studying people with Mosaic Down syndrome for around 40 years now. So we have, if you look at our population and years of follow-up, about a fourth of our individuals, we have at least 25 years of follow-up on these individuals. So it's a really robust data set to try to understand changes associated with aging. If we look at our age distribution, again, about a fourth of our individuals are at least 30, 33 or older. We also have young individuals as well as nice distribution across the lifespan, so all ages. And again, with many of those individuals that we have, at least 15, more than half of our individuals, we have 15 or more years of follow-up. So a rich database to understand aging. I don't know if Katie's still on or not. But one thing we're hoping to look at from the longitudinal study design is you're looking at patterns that we see when someone's a child and then following them across at least 3 different time points and seeing what are the changes that occur as they go from child to adolescence to an adult. And so we look within a time point at the differences between the trisemic and disemic cells, and then between time points, looking at the differences in the trisemic cells at, let's say, a child versus an adolescent versus an adult. Also looking at the changes in the disemic cells across those timelines. So this is what our current research grant is looking at. One area we've been focusing on is a process called senescence. This is a normal biological process that starts from embryogenesis on. It's one of the processes that leads to the typical patterns that we see of organ development and embryogenesis, but can also be associated with aging and cancer. On the bright side of senescence, so the sort of the good side, the bright side, it can be help with tumor suppression. It can prevent fibrosis. It's involved in normal development. It can assist with wound healing, immune cell attractions. But it does also have some challenges. So on the dark side, it can impair regeneration. It's associated with aging. It's also associated with chronic inflammation. It can lead to differences in cell proliferation that can be associated with cancers, particularly leukemias, but can also be involved with solid tumors. So this is a process that we're really interested in learning more about for people who have Down syndrome or Mosaic Down syndrome in particular. And so this is one of my graduate students, Tori. And so we're looking at a way of measuring senescence. It's called SADS. It stands for senescence dissociated distension of satellites. We tried the system out on cells that were known to have senescence in them. And as we would expect, this is a very good reporter for detecting senescent cells. We then went on to try it in our individuals who have Mosaic Down syndrome, looking at their disemic compared to trisemic cells. This would be an example of a SADS pattern. This would be a typical pattern that's compact. And so when we look at individuals across different ages that are on the x-axis and looking at the proportion of cells with the senescence marker, what we see is that overall, we see a higher percentage of the senescence marker in the trisemic cells, shown here in orange histograms, compared to the disemic. But it's not, not all people, so there's some variance. In general, when we look at senescence-related markers, we see a statistically higher frequency in trisemic compared to disemic cells. So this could be something that, we talked about the percentage of changes in blood early on. So if I have a person who has 40% of their cells that have a trisemic change, if I take this cell and make it senescence, that means it doesn't divide anymore. It can still stay alive, it's just not dividing. So then when those cells continue to divide, if each of them have, make a double of itself, then this change in percentage over just 2 divisions can go from 40% down to 27%. So this senescence may be part of why we're seeing changes in blood, which is an inflammation-related type tissue, compared to other tissues. So for example, if we see the brain, we said we don't expect to see most of the brain cells, we don't expect to be dividing. So we would expect, even though the blood cells, they all started at 40%, we might see the blood go down to 27%, but the brain might be still at 40%. Or we might see some changes, but less senescence effects. So we might see a more modest change. So this is something that we're also looking at, is trying to correlate those. And then, is this a predictive marker for someone who's at risk for developing some of our aging-related traits that we see. We're especially excited about this study because some of the senescence markers actually have medications that can be used. And so these senolytics are actually ways that you can treat or reverse the impact of senescence. And so this might provide a means of alleviating some of the early aging conditions that we see in people with Mosaic Down syndrome. Other areas that we're looking at a measure of chromosomal instability that's called a micronucleus. This is the general term is called cytosolic DNA. This is what it looks like at the microscope when you're looking at it. The cell divides, and then some chromosomal material can actually be excluded or kicked out of the cell, if you will. This can then be detected as DNA that's not where it's supposed to be, and then that can trigger inflammation changes. When we go on to look at these, these micronuclei or cytosolic DNA have been associated with a wide variety of conditions, including neurodegenerative diseases, including Alzheimer's, gastric problems, skin diseases, psoriasis, inflammation, as well as aging. So there's a lot of overlap between the conditions that these have been associated with and some of the traits that we see that are acquired with aging in people with Down syndrome. And this is just an example. And you'll see I've had pictures of some of our participants who have submitted their pictures for our annual IMDSA drive to increase awareness. So thank you for sharing those photos, and I hope you don't mind me adding them to make the talk more interesting. This is a schematic that shows an example of a chromosome that didn't sort correctly. It got sort of kicked out or left out and then was excluded out. In this case, this was a chromosome 21. So you can see one of the daughter cells still has 3 21s. This daughter cell has only 2 21s left. The third got excluded. So this cell, initially was trisomic in both cells, but now we have a disomic cell that's formed. This is an example where these changes can involve other chromosomes. This would be a chromosome 8 that was excluded. And sometimes it's neither an 8 or a 21, which are the two probes we're looking at in this example. And so when we compared, again, across multiple ages, what we see, if you look at the trisomic cells now shown in blue compared to the disomic, we don't see a lot of difference in the earlier years, but as individuals get older, we see a significant increase in the frequency of micronuclei in trisomic compared to disomic cells. So this is something that we think might be helpful as a biomarker for someone who might be at risk for developing some of the age-related conditions. When we looked longitudinally, so now looking at individuals where we have multiple time points, and in parentheses, it shows the ages, like this was, the first sample was collected at age 7, the second at age 10. This is one collected at 35 and 41. And so we see a similar trend also within an individual, and that as an individual gets older, they tend to have a higher frequency of micronuclei, actually statistically significantly increased. We're also looking at another aging hallmark that's called a telomere or subtelomeric region. So these are associated with aging. When we look at telomere lengths in trisomic cells, they're shorter compared to the genetically identical disomic cells. And this is what we call a heat map. It's much like the maps that you see on the weather where you have strong rain or light rain. These show short telomeres in green hues versus longer telomeres in red hues. And when you look at the map overall, you can see that most telomeres are shorter. What we've done is to compare the average in the trisomic cells to the average in the disomic cells. And so that difference is shown here. And you can see that in general, the trisomic cells are statistically shorter than the disomic cells. We're also developing a new telomere assay that is more robust than any of the other assays that have been done in the past. This is work that's been developed primarily by one of my former research members, Dr. Lilia Gaggiani. She's someone that I think will likely continue my research into the future as I'm sort of preparing her to hand off some of the research to her as she continues on to get grant funding and keep the projects going when I at some point decide to retire, not immediately. But anyway, we've got DNA that we can prepare using special methods. We can use what's called an atomic force microscope, and then we can actually measure molecules and get very precise estimates of telomere length. Currently, I have two NIH funded grants. We've had one of these was a K08 that Ruth Brown, that is also working on research related to Mosaic Down syndrome. I have the work with the aging grant. We got a grant that also was for one of the conferences related to Down syndrome. And then I have another grant that's looking at aging in twins. So that's another NIH funding that I have. So at this point, let me see if there are other questions.
It looks like we have one more.
Okay.
It's down at the bottom and it says, thanks so much for all the research over the last 40 years. What made you interested in researching Mosaic Down syndrome?
Good question. When I started into graduate school, my PhD was done on Down syndrome, so non-Mosaic Down syndrome. And I chose that as a project because it is the most common chromosomal condition. And so it seemed like research that could have an impact, make a difference. And in doing that research, has studied a few individuals who had mosaicism and learned that most of the time people who have mosaicism actually have two chromosome sorting problems that give rise. They start off trisomic and then have a second sorting problem that gives rise to the disomic. So it seemed like by studying people with mosaicism, you could actually learn more about what are the factors that contribute to having chromosome sorting issues. And so when I started looking for people who have mosaicism, it wasn't easy. I sent letters out to every Down syndrome support group in the nation, and got people who, I started initially through the common routes of asking geneticists to refer, and really had hardly any referrals. So I decided I'll just take it to the families directly. And then people started contacting me and talking with those families. Had the common comment, nearly every family was like, there's nothing out there about mosaicism. There's one sentence in the Down syndrome book, and there was absolutely nothing out there for families. And so I decided this was what I needed to do. And, this was an area in need of a champion, and an area in need of information, but also an area that's extremely rich for helping us to understand what's going on with Down syndrome. Among geneticists, there's a phrase that I was taught when I was trained. It's like to cherish your exceptions. And I think that's really what people with mosaicism are. They're while less common, they should be cherished because the amount of information that you can gain by comparing the genetically identical trisomic and disomic cells is much, much, much stronger than the information that you can get from studying unrelated individuals. So that's what instigated me. Other questions?
Doesn't look like it.
Well, thank you all. I know this went probably a little longer than you were expecting. Brandy, any closing words?
No, but just thank you for, all of your expertise, Colleen. We do appreciate you and all of your time and energy that you put into this, even on your Sunday afternoon, spending it with us to educate our families. So we do appreciate that. In closing, I'd like to just remind you guys that we are heading up for a big fundraiser that's going to be taking place in March, and it's our awareness campaign. And it's the one where you saw where Colleen was using all of those photos. We share those on all the social medias, but we also allow for families to make their own personal fundraising pages. So hopefully if you guys feel the need and want to help contribute to, what we do here at IMDSA and further research and then making our community stronger, I would love for you guys to participate and help us kind of continue to build on that. So just keep an eye out on the newsletter. We'll probably go out typically around the 15th of the month, which will be next month. We'll have that newsletter ready to go out, and there'll be more details on the fundraiser then. So, but in closing, I don't have anything else unless anybody else has anything. And she wrote back, thank you. See you from Australia. Nice. Okay. And Colleen, also, if you guys check the chat box, she just put her direct email in there. So if you guys need to get her in direct contact with her, you can just email her directly. So again, I appreciate everybody taking your time today. Thank you so much.
And I see there is a question that just came up where someone missed the beginning. Yes, we have recorded the session and we do plan to make that available to people. The details we're still working out. So we will share the details as soon as they're available.
Yes. We'll most likely put it on our YouTube channel. We don't, we do have a small YouTube channel, and as we get videos and stuff, I don't think we'll be able to email it directly to you guys. It'll have to go up on that YouTube channel, and we'll probably share. I would think it'll probably take us about a month. Nick, who is our treasurer, he will be the one that's going to help us get that done. So he's also works and he's a teacher. So I'd like to give him at least 30 days to try to get that conquered. So give us a little bit of patience. And like I said, hopefully in 30 days, we'll be able to send this out with a link to directly to that page. But again, thanks everyone for coming. All of y'all's questions were so good. And I appreciate all of the questions too.
And just feel free, if you would like to contribute, if you'd like to join our research study, you can feel free to reach out to me directly. If you just have a question, that's fine. And, research isn't for you right now, but you have a question, feel free to reach out. Also, I will make one last plea. Wearing my IMDSA scientific advisor hat. This is a group that's 100 volunteers. And so I'm seeing a lot of talented people on this session. And so, it doesn't have to be a full-time job commitment, but if you have some hours that you can help, we would love for people to help. There are a lot of different roles that you could play, different things that you can do. So if you're inclined to help out the organization, that would be wonderful. And you can talk to Brandy directly. And I'm sure we can find a niche that we would benefit from having talented people that are on the line right now. So thank you for joining us, and feel free to reach out at any time.
Thank you. I appreciate that. And then within closing, it looks like Amanda has a question. If you want to have time to answer that, if not, we can maybe do that offline.
As far as so there's a question in the chat from Amanda, who's a nurse and works with many new drugs, and asking if biologics have been studied in inflammatory issues related to Down syndrome or Mosaic Down syndrome. And the answer is yes. There's actually a clinical trial that is underway. It's through folks at the University of Colorado, and they have had some really good success with some aspects of inflammation, in particular hair loss, alopecia. They've had great success with clinical trials there. So this is an area. You can also go, there's a website that they've redeveloped as part of the NIH funding. But if you go to Down Syndrome Connect, so DS-Connect, CONNECT, it has a list there of all the different clinical trials that are available for people who have Down syndrome. Some of those allow people with mosaicism to join, some unfortunately do not. And so that's something we continue to work on from an advocacy standpoint. The reason being just statistical challenges that they have. But you can look at the trials, and even if it says that they don't take mosaics, it wouldn't hurt to at least reach out to them and ask.
Yes. I always say, if you're interested in research, always let them know ahead of time that your child does have a confirmed diagnosis of mosaicism. The only reason is I hate for people to go in and take time with these research studies and then take part in them. And then they come back and say, oh, your child has mosaicism. Unfortunately, you don't qualify. So always let them know, because when it comes to research, I guess it obviously plays a big part into the outcome of the study. So always let them know. Any other time, it really doesn't matter, but when it comes to research, it does.
And then if this is something that you found helpful or new directions you'd like to go, be sure to share those thoughts with Brandy so that we can be, we want to meet your needs. What's helpful to you? What are the sorts of questions you'd like answered or the sorts of information that you'd like to hear for future sessions?
Yep, our next one is going to be with Dr. Ruth Brown, and that is on January 28th, I believe it is. It's on a Wednesday, I believe. And that one will be basically just on her past study that she did with feelings and, depression and anxiety and with individuals with Mosaic Down syndrome. So hopefully you guys can pop in and join us for that session. Okay.
Okay.
So Colleen and I will get with Nick and we will get with you to try to get this up sooner than later.
Okay.
I did talk to him and he said, I think that's where we're going with it. We're going to put it on the YouTube channel. So.
All right. And if there's anyone there that had photos that I used that were, public domain
It's
photos.
part of our thing. They had to sign a thing, so it's
All
not,
right.
You're fine. They signed it and it's part of it. Like we own it. So yeah, it's fine.
Okay. All right.
You're good.
So I'm good. Okay.
Yes.
Just wanting to make sure I preserve confidentiality.
Yeah, you're fine. So.
All right. Thanks, everybody.
Thank you.